Hello, this is Mrs. Gabrick. We're going to learn a little bit about gene linkage today, and we're going to follow along with this PowerPoint called Make a Life to Save a Life. Before you start this video, you need to go onto Moodle, which is probably where you're watching this, and there is an online quiz. And you're going to be toggling between this video and that. So to begin, find the online quiz that goes along with Make a Life to Save a Life, and then do the first 8 questions. Those will review meiosis and mitosis before we begin this. Once you've completed those, come back to this video. So pause your video, go do the quiz, the first 8 questions, and then come back. Alright, welcome back. So the case study here is about Jack and Lisa Nash's daughter, Molly, who was born with a rare, incurable genetic condition called Fanconi anemia, which will render her body unable to produce enough blood cells. To find a correct match for tissue transplantation, which is what's going to be necessary to keep Molly alive, really depends on matching a specific group of proteins called HLA for human leukocyte antigen. These are found on the surface of white blood cells that are used to distinguish one's own cells from foreign cells. So in the picture up here, you can see that we have a white blood cell there, all right? And on the surface of that is a protein. HLA proteins are encoded by several genes on chromosome 6. There are many variants or mutations of these genes, and these are called alleles, which you already know. All right? So you can see to the picture on the right or left, we've got HLA A, B, C, DR, DQ. We've got quite a group here. All right? There are lots of different variations of this. Look, for A, there's 697. B has even more. E only has 9. When a white blood cell called a macrophage eats a foreign cell, it uses these HLA proteins to display the foreign cell's proteins. So this big blue guy here, the macrophage, will gobble up a foreign cell, could be a virus or bacteria, and then on its surface, there's some different HLAs here, and after it gobbles up that invader, the lysosome will digest it, and then it will put the proteins onto the HLAs. Shoot, I got to start again because I didn't make mine. Hi, Mrs. Gabrick here. Welcome to the video, Make a Life to Save a Life. Before you begin this video, on Moodle, there is a multiple choice questionnaire or quiz that goes along with this. And what I want you to do is do the first 8 questions to help you review mitosis and meiosis. When you're done with those 8 questions, come back to this video and we'll continue. So pause your video now and go and do the first 8 questions. Welcome back. The case study we're going to be looking at is about Jack and Lisa Nash's daughter, Molly, who was born with a rare, incurable genetic condition called Fanconi anemia. This renders the child's body unable to produce enough blood cells. When we match organs for tissue transplantation, we look at the HLAs, which stands for human leukocyte antigen. These are found on the surface of the white blood cells, and that's how you're going to distinguish a foreign cell from your own cells. So up here you see a white blood cell, and there on the surface over here are the proteins in the cell membrane. HLA proteins are encoded by several genes on chromosome 6. There are many variants or mutations of these genes called alleles. So you can see gene A, or HLA-A, has 697 alleles. B has even more, whereas E only has 9. When a white blood cell called a macrophage, seen over here in blue, it eats a foreign cell, could be a bacteria or virus, it takes the HLA proteins and uses them to display the foreign cell's proteins. So it gobbles up the foreign cell, puts it in a lysosome, and then displays the proteins from the foreign cell on its membrane. That's going to help your immune system attack and destroy these foreign invaders. HLA proteins, like anything on your genes, are inherited from your parents. So Mrs. Nash is diploid, as are all humans. So she has 2 homologous chromosomes, 6s. All right? Take a look at this. Is she homozygous or heterozygous? Hopefully you said she's heterozygous because she has 2 variants. She has HLA1 and HLA2. For the B, she's got B35 and B44, so she's heterozygous for both of those genes. Okay, so now, in a second, you're going to go back to your online quiz and answer this question. So based on the picture of Mrs. Nash's white blood cell, is the inheritance of HLAs, and there are your 5 choices. Answer that question, pause this video, answer that question, and then come back. Alright, so a quick review of mitosis. You know it's used in asexual reproduction. We don't need it to make sex cells. It occurs in your body cells or somatic cells as you grow and divide. The cells made by mitosis are genetically identical. And you can see that the cell we start with here is exactly with the 2 cells that we end up down here. This is not a way to make new traits for reproduction, okay? If you want to do sexual reproduction, you need a different method, and that's meiosis. So, diploid organisms need specialized sex cells. They allow the traits to be combined from the 2 different organisms, mom and dad standing there, okay? You can't fuse any 2 random cells. It's got to be sex cells. So we've got to go through meiosis, make those sex cells, put them together, and then our offspring will be diploid again. So meiosis is that specialized cell division so that you only have 1 of each chromosome, or your haploid. These gametes, or sex cells, are made only in the gonads. In a male, the testes, in the female, the ovaries. Here's a comparison between mitosis and meiosis. Hopefully you remember the basics of this, but if not, take a second, pause it, and review the diagram. Keep in mind, during prophase and metaphase, we're going to differ in mitosis and meiosis. In metaphase, when they line up, they do not line up as homologous chromosomes. Remember, these 2 are chromatids. Okay? Here, we have a tetrad because we have our 2 sets of chromosome 6s, or our homologous chromosomes, together. This close proximity does not favor crossing over if the genes are very, very close. Okay? But again, in the tetrad phase, crossing over can occur, as you see here. Okay, so there again we see mitosis compared to meiosis. Because of the way they line up in metaphase of mitosis, or metaphase 1 of meiosis 1, what we're going to end up with is something different at the end of each of those events. In mitosis, we've separated the chromatids. In meiosis 1, we've only separated the homologous chromosomes. Homologous chromosomes, therefore, in meiosis 1 are separated into 2 different cells. Each new cell has only 1 of each different chromosome, so they're haploid. Now, we still need to separate the sister chromatids, so that the total amount of chromosomes in DNA is truly half of a normal cell. That's what occurs in meiosis 2. In meiosis 2, we separate the chromatids. Then we end up with 4 haploid cells. Okay, so pause your video, go back to your quiz, and answer this question now. You can see on the right Mrs. Nash's chromosome 6s. Okay? When she makes eggs, what are the possibilities for the alleles in those eggs? Here's question 11. There's Mr. Nash on the right. You can see his chromosome 6s, what types of sperms could he make? Oops, that should just be question 12, not 812. Looking at the combinations, which of the following shows one combination of HLA-A and HLA-B genes expected in an offspring of Lisa and Jack Nash? 13. Is it possible for any child born to this couple to be identical in both HLA-A and HLA-B genes with either parent? 14. Using the proportion of each type of sperm and egg that you calculated in questions 2 and 3, what's the likelihood that Molly's sibling would inherit the same combination of HLA genes and thus be a good organ donor for Molly? By the way, at this point, you've probably realized we have a bird at home, so I apologize for the bird singing in the background. So clearly, Molly's best chance of finding a bone marrow match was with a sibling. Unfortunately, Molly's an only child. The Nashes always wanted to have more children, but because Fanconi anemia is an inherited condition, they knew that if they had another child, that child had a chance of getting the disease just as Molly had. But neither Jack nor Lisa had the disease because the mutation is recessive. In order to have Fanconi anemia like Molly, both copies of the FANCC gene would have to have this recessive mutation, and they only had one. How is that possible? Well, think about what we've done in the past. If Mr. and Mrs. Nash are like this, their child Molly could have gotten a little left from Mom and a little left from Dad. So here's what it looks like. FANCC for Fanconi anemia is on chromosome 9. Now, again, we've got homologous chromosome 9s in Mom and in Dad, but only one of those carries the recessive. So here we've got the chromosome carrying the normal gene, and this chromosome carrying the allele for the disease. Dad in the same situation. This is why Molly ended up being the right. All right, question 15. Using what you know about meiosis, how meiosis created eggs and sperm, what is the likelihood that Lisa and Jack could conceive a sibling from Molly that would not have Fanconi anemia? You can do the Punnett square to check that out and see what the possibilities are. In this case, we're only worrying about Fanconi anemia, so we're only looking at chromosome 9. When they discovered that they could conceive a baby that was free from Fanconi anemia, Jack and Lisa underwent in vitro fertilization, followed by a procedure called pre-implantation genetic screening, to choose an embryo that would have HLA proteins B44, B35, A2, A26 that matched Molly, and to choose an embryo that would also be free of Fanconi anemia. Now, there are ethics in this. This is the first example of using the pre-implantation genetic screening to select a baby solely to treat its sibling. There are going to be ethical objections. You could think of reasons why they should be allowed to do this and what sort of regulations. So just think about these ethical questions. All right, select a baby. There's how Molly ended up. Question 16. Using your knowledge of how chromosomes segregate, this is now a dihybrid, what percentage of Mrs. Nash's eggs would carry a chromosome 9 and a chromosome 6 with the A2, B44 alleles and the normal gene? This is what it's going to look in a dihybrid cross, where we're looking at the two possibilities. And keep in mind, they could line up differently. Question 17. If the same percentage of Mr. Nash's sperm carry the correct chromosome 9 and 6, what is the probability that a single embryo would be a perfect HLA match for Molly and not have any alleles for Fanconi anemia? That's a big Punnett square, but you can use that or you can do your own. And there's the update on the Nashes. I'm going to let you pause your computer and read that, because I'm almost out of time. All right? Make sure you finish the quiz, submit it,